Hi, science friends. Let's take just a minute to revisit what we've learned in the last couple of periods. So, what are the three stages of cell signaling? You should know those like the back of your hand: reception, transduction, response. Those are the three stages of cell signaling. Reception starts when the molecule binds. Transduction is those relay proteins in the middle, the series of steps, and then the response is whatever the cellular response may be. Local types of signaling, you might say paracrine, you might say juxtacrine, you might say autocrine. You may also say like synaptic signaling. That would be the nerve cell signaling. Long distance signals in the third question are going to be sent through the blood, through the endocrine system, generally through hormones. Okay? And then the last question, the three main types of transmembrane receptors. You should have the ion channel receptors, the G protein coupled receptors, and the receptor tyrosine kinases. Those are the three main types of transmembrane receptors. So if you struggled with any of those are very basic questions. AP test questions would be a lot more deep than that. Then you're going to need to go back and revisit this material. So remember, that's critical to doing well, is just being on top of it every single day. Okay? So without further ado, let's go ahead and jump into today. So I would recommend today we are going to talk about 4 major enzymes that have activity in signal transduction pathways. And I have in my hand a note card that I've made with these 4 enzymes. And I would suggest that you do the same. There are only 4 of them, and so you think they're not that confusing, but they have kind of long names and they do the opposites of one another. So they are very confusing. So I would recommend you pull out a note card or a small piece of paper and make this, just write it down so you can reference it quickly and easily. I have to review these at the beginning of every year at this unit. I've taught AP for 5 years now, but I still have to review it at the beginning of every year and always have my note card handy. So the first enzyme that I want to talk about is protein kinase, and it's probably the easiest one for me to remember for some reason. But kinases, all enzymes, remember, are going to end in ACE. Anything that ends in ACE is an enzyme. And then usually what they, or not always, but oftentimes what they act on, if it's a sugar, it's going to be the sugar that ends in ose. For example, lactase is the enzyme that breaks down lactose, the sugar. Galactase, the enzyme, breaks down galactose, the sugar. Okay? So that's not going to be always the case, but with sugars, that very commonly is the case. So we're not going to really see that example playing out perfectly for us today, though, unfortunately. But protein kinase is the enzyme that transfers phosphates to a protein, and those phosphates are coming from ATP. So remember, we've talked about the structure of ATP before. You've got that adenosine molecule, and then you've got the triphosphates, okay? And so when one of those is cleaved off, energy is released, and this phosphate can be transferred to something else, okay? I don't know. It's going to be transferred to something else, and generally that will become activated. Okay, I'm kind of sloppy here. That will become activated. But the enzyme that is responsible for this transfer is protein kinase. Okay, transfers that phosphate to the other molecule, which often activates that molecule, represented in my fancy diagram here as a star. Okay. And so that process of transferring that phosphate is called phosphorylation. We talked about this a little bit last week when many of you all were in there with me on Wednesday and Thursday. So now we said generally it's going to activate the protein. I want you to know, though, it's not always going to activate the protein. It may deactivate the protein, okay? What it is always going to do, though, phosphorylation is always going to change the shape, okay? So when something binds, it's going to cause a shape change. So while generally we see that causing an activation, it can cause a deactivation also. So let's have two examples here. Okay, if you have an inactive protein and it gets plus a phosphate, that's going to activate the protein generally. If you have a active protein and you add a phosphate to it, it is generally going to deactivate the protein. Okay? Because it causes a change in shape. Remember, shape determines function. And these proteins bind to something else or they have a job. And if you change their shape, they can't do their job. Okay? And so if it's binding to an inactive protein, it very likely is going to change its shape to allow it to do its job. If it's binding to an active protein, it's very likely that it's going to change its shape to make it not be able to do its job. Okay? So for the most part, what we're going to talk about, it's going to activate the protein, and that's what I taught you all last week. But I want you to know there are some exceptions to that rule. But binding of something is always going to cause a shape change. Okay? That's always going to be true. All right, so here we've got a phosphorylation cascade. So in this phosphorylation cascade, we're adding phosphates, which is going to activate, in this case, these proteins. So you see here, ATP is converted to ADP. So it's lost that phosphate, okay? And then that activated this protein, okay? In this one, ATP is converted to ADP. And so that protein activated this protein, okay? And then the same thing, so on and so forth. ATP is converted to ADP. That phosphorylated that protein, which activated another protein, which went on to cause our cellular response, okay? So this piece of it that has all of those phosphorylation steps, and you see that phosphate, where did it go? It bound onto here, and it caused a shape change. We had this oval, okay? And when that phosphate bound, we had this nice circle, okay? So these are obviously very simplistic drawings, but you see the phosphate being transferred from the ATP to from the ATP to the inactive protein kinase, causing it to be activated, okay? And so that is the phosphorylation cascade. Cascade just generally means like series of events or steps or whatever. It's kind of a fancy word for something that's not super fancy, okay? So these were activated when the signaling molecule bound, okay? So this is our first step in our signal transduction pathway. Reception, all of this here in the middle is going to be the second step, which is transduction. And then here at the end, we have our last step, which is the response step. So the signaling molecule binds. Now remember, it is outside the cell in this case. This is our transmembrane receptor, which could be a receptor tyrosine kinase, a G protein coupled receptor, a ligand or an ion channel, any of those, a gated ion channel. And our signaling molecule binds, and that signal is sent from the outside of the cell inside the cell, okay? And so once that receptor receives that signal, then it is going to activate a relay molecule. We are going to talk about this relay molecule in just a second, and we are going to call it a second messenger, okay? These second messengers go on to activate these protein kinases. So this second messenger here it's a molecule, it's not a protein, okay? A molecule is very different from a protein. Things like water and ATP and carbon dioxide and glucose, Those are molecules. They do not have the structural, the amino and carboxyl and the R group that proteins have. They're not made up of polypeptide chains like proteins are. So it's a molecule, not a protein. But these relay molecules, or these second messengers, kind of a different word here, are known to activate these proteins, like this one, like protein kinase 1, which cause this phosphorylation cascade to take place. Okay? And then ultimately lead to our cellular response down here. So this picture is really important. We can get a lot out of this picture and being able to explain this. Okay? Now, I'm going to come back to this picture in just a second when we talk about this next enzyme. So the second enzyme after protein kinase that you might add to your note card would be protein phosphatase. So it has the opposite job of protein kinase because it is involved in dephosphorylation. So it removes the phosphates, okay? So generally, like we said, not always, generally it is going to inactivate those proteins by removing the phosphates, which would help turn off the signal pathways, okay? And then those protein kinases can be reused. These proteins aren't used up. They participate in the reaction, but they're not used up in a chemical reaction, and that's really important. So we see these protein phosphatases. Oops, if I can get back to my diagram. I deleted all my writing, but that's okay. That's maybe good. So right here, we see these protein phosphatases, and their job is to try to convert these activated protein kinases back into the inactive versions by removing that phosphate, like we just talked about, by dephosphorylating, okay? And you see them in every step of this phosphorylation cascade, okay? That protein phosphatase right there. That's what that PP stands for. And so in the process of doing that, it inactivates the kinases, okay? And so these kinases go back to this inactive form, all right? And they're going to remain in that inactive form until they receive another signal, which activates the relay molecule or the second messenger, which we'll talk about in just a second, and causes this phosphorylation cascade that leads to our cellular response. Okay? So there is our first two enzymes that we're talking about. Remember, they're all going to end in an ACE. That's going to be an enzyme. Okay? So we're going to talk about two second messengers now. So there's lots of second messengers, but the ones that we're going to talk about today are cyclic AMP or CAMP and calcium ion. Okay? These are our second messengers. So this is where they are. Okay? This is kind of where a second messenger might be. You can see second messengers, though, anywhere in this pathway. What you may want to write down in addition to here, second messengers are non-protein molecules involved in the signaling pathways. Okay? It doesn't really matter where they are. This one makes it look like it's right here at the beginning, so you would call it like the second messenger. But if you had it down here somewhere, it could still be a second messenger and be involved in a very late step in the pathway. So long as it's not a protein, we can still call it a second messenger. I know that's kind of counterintuitive, but that's the way that they name them. A non-protein molecule or ion that's involved in a signaling pathway. Okay? It's going to be inside the cell. And so, CAMP is the first second messenger we're going to talk about. Calcium ion is the second messenger that we're going to talk about. And so CAMP, or cyclic AMP, which stands for cyclic adenosine monophosphate, ATP is converted to CAMP by an enzyme known as adenyl cyclase. So that's the third enzyme that you might want to add to your note card, adenyl cyclase. And its job is to convert ATP to CAMP. And that's adenyl cyclase. Okay, notice it also ends in ACE. Okay, and so you see it here in this diagram. We have ATP right here, our triphosphates, and then our adenosine. Okay, and we have in our reaction arrow, we have adenyl cyclase. Enzymes are always written over the arrow in a chemical reaction because they are not a reactant or a product. They're not used. They're not part of the reaction. They just help to facilitate or speed up a chemical reaction. We'll have a whole unit dealing with enzymes in the next unit. So adenyl cyclase does that job. It converts ATP to CAMP, okay? And what you see, if you look at the structure of ATP in CAMP, ATP has these 3 phosphate groups, okay? CAMP only has 1 phosphate group. So you might say, Ms. Brown, why isn't that just AMP? Why isn't that just adenosine monophosphate? Well, it's cyclic, and that's what that C stands for. Because of this bond right here that kind of pulls the structure into a ring, so to speak, that brings it in here and makes it more one with the adenosine, okay? And so that's CAMP. So the molecule, or the enzyme that does that is adenyl cyclase, converts ATP to CAMP, okay? Now, a second enzyme, or fourth, if you're writing these on your note card, is phosphodiesterase. And phosphodiesterase is responsible for converting CAMP to AMP. And so essentially it's going to just break that bond right there, and then this is going to look more just like an adenosine monophosphate. It's not got that cyclic portion anymore, okay? And so the second messenger here is CAMP, okay? And CAMP is really important to be able to convert ATP into AMP. And then AMP is something that will go on and help trigger protein kinases to continue the phosphorylation cascade in the signal transduction pathway. Okay, these second messengers help with the continuation of those cascades or of that transduction step in the signal transduction pathway. Okay, and that's what this says here. CAMP usually activates protein kinase A, which phosphorylates other molecules in the pathway. Okay, so here's our second messenger. We've got the first messenger, which is our signaling molecule. That's always going to be the first messenger. It'll activate the G protein. Okay, this is in a G protein coupled receptor example here, which activates adenyl cyclase. That's that first enzyme. Adenyl cyclase is going to convert ATP to CAMP, our second messenger, and then CAMP goes on to activate protein kinase A, which elicits a cellular response. And so you see there our second messenger, CAMP, that helps in this pathway, that causes that phosphorylation cascade. Okay? And they're not always going to be drawn in the diagrams. Well, I guess you see it there. That was our second messenger. Okay, it activated this protein kinase here, which went on to start the phosphorylation cascade. Okay. The other second messenger I want to talk about is a calcium ion. So calcium is a positively charged ion. And so if you notice here, it says calcium pumps actively transport calcium ions from the cytoplasm out of the cell or into the ER. So thinking back to the last unit about active transport, what does active transport mean? Well, it means two things. It means that it requires energy. So here where we see our calcium pump, you see that energy is being inputted in the form of ATP. But it also means that it transports from a low ion concentration to a high ion concentration, okay? Normally, passive diffusion or passive transport is high to low. Active transport is low to high. And you'll see that down here if you look at the key. The blue, which is in the extracellular fluid, is high, and then the orange or peach in the cell is low. And that's what active transport is. So the calcium ion is a second messenger that can help to do this, okay? It's really important in muscle contraction. We see calcium ion to be critical in that capacity, okay? One other thing I was going to tell you about camp here, going back for just a moment, speaking of muscle contraction. So has anyone ever heard of the disease or the illness cholera? You don't really hear it around here or in, it's like third world country kind of thing. But cholera comes from water that has been contaminated with feces. And it makes people really, really sick. And they can often die from cholera. So essentially what happens in cholera is a bacteria that produces a toxin, and the cellular response of the toxin is to cause salt to be pumped into the intestines. Okay? Now, you have salt, large amounts of salt, being pumped into the intestines, okay? The cholera toxin activates a pathway that has this response, okay? But what the toxin actually does is it affects the G protein coupled receptor by causing it to not be dephosphorylated. So in other words, this pathway stays on. Sometimes that's normal. Sometimes salt gets pumped into the intestines, and that helps water move into the intestines, which that's good because that helps you poop and go to the bathroom, okay? And not be constipated, okay? So that's normal. We want that to be on all the time. But what we don't want is this GTP to be stuck there and unable to be dephosphorylated. And that's what happens with cholera. So this pathway gets stuck in an on position, okay? And so salt continues to be in high levels in the intestines, okay? And so thinking about what we know from osmosis, if there's a high level of salt in the intestines, where's water going to try to go? Well, water's going to try to go into the intestines, okay? All the water from the body is going to pump into the intestines. So your body's going to become very dehydrated, and you're going to continually, your bowel movement's going to be liquid, basically, okay? Everything's going to run straight through you, and you're going to be seriously dehydrated. You're not going to be able to absorb or retain any water. And people actually died. Lots of people died from cholera before we had water purification systems and stuff that we have today. So treatment for cholera is about being able to deactivate this G protein coupled receptor, okay? Putting medicines in that will cause this signal pathway to be turned off. So there's just a little bit of an application for that second messenger in the example with cholera, okay? Other medicines work with these receptors too. For example, heart medicines that help dilate the blood vessels that lead to the heart work with G protein coupled receptors, and they help to enable this pathway that causes muscles to dilate by phosphorylating those proteins. And that's really important for people that have constricted heart vessels. So, sorry, back to this second messenger. Those calcium ions is going to allow active transport from the cytoplasm out of the cell, or we also see them here into the endoplasmic reticulum. Okay, again, active transport from low concentration to high concentration of calcium ions. Okay, and so our second messenger, if you look at it here, it looks like it's actually pretty far down in the pathway. You may say, Ms. Brown, that's not the second messenger. Look at all of these molecules that are involved here before we get to this second messenger, this calcium channel, okay? I don't really know. So all of these are considered to be second messengers, okay? This DAG is a second messenger. This PIP2, whatever these are, I'm not really familiar with this. As long as they are not proteins, okay? We are really looking, though, at this calcium ion as a major second messenger, okay? Calcium ion's probably more popular and more widespread than the CAMP, excuse me, okay? But there's others as well, okay? We're just not talking about them. We are talking about CAMP and calcium ions as our second messengers. Even though CAMP is several arrows through the pathway, because it's a non-protein and it helps stimulate other proteins to elicit a cellular response, it is considered a second messenger, okay? So I think that covers everything we need to know today. Tomorrow we'll get into cellular responses a little bit more. Today you're going to be working on part of the handout on signal transduction pathways, and we'll come back and finish up the rest of that tomorrow. Y'all have a great day.